Good morning. My name is Divya Pradhan, and I am going to talk to you about the histology of the nervous tissue. The nervous tissue is a highly cellular and made up of two types of cells. The neurons, or the excitable nerve cells, right here, and the neuroglia, or simply glial cells, or the supporting cells. We are going to talk about the neurons in detail. For now, let me tell you that the neurons are the ones that will transmit the information to the brain and away from the brain. The neuroglial cells, or the glial cells, are the supporting cells, each with unique shape and function. They will be discussed later on in the other integrity clips. Neurons are the basic building blocks of the nervous system. Neurons are also called nerves and are specialized cells and have very unique characteristics that include they conduct nerve impulses, or in simple words are responsible for receiving and transmitting information. Each part of the neuron plays a role in communication of information throughout the body. They have very long, extremely long longevity and can survive for over 100 years. So basically they are built to last for a lifetime. They are amitotic, that is they lose the ability to divide. So if they are damaged, they cannot be replaced. The exception is the hippocampus, should be named, which is involved in memory that can regenerate new cells. They have a very high metabolic rate and therefore must have a rich supply of mitochondria. Now remember, mitochondria is the one that generates ATP, but only in the presence of oxygen. So these nerve cells need to have a rich supply. They cannot survive without O2. The neurons have two main body parts, the cell body as well as the processes, dendrites and the axon. The neuron or the cell body is also called, the neuron cell body, is also called soma, and it consists of the spherical nucleus as well as a nucleolus. Can you see this? The soma and the nucleus do not play an active role in the transmission of the neural signal. Instead, these two structures serve to maintain the cell and keep the neuron functional. The support structures of the cell include mitochondria, as we mentioned before, which provide energy, Golgi apparatus, which of course is responsible for the secretion. The rough ER is extremely well developed, and these are called the Nissl bodies in here. They are thought to be involved in the synthesis of neurotransmitters. These neurotransmitters are basically chemicals with which using this, the neurons will communicate with each other, as we'll study later. Note that the cluster of cell bodies are called nuclei. This is in the central nervous system, and they are called ganglia in the peripheral nervous system. There are arm-like processes that extend from the cell body, as we said. One of them is the dendrites and the other one is the axon. Bundles of neuron processes are called tracts in the central nervous system, and nerves in the peripheral nervous system. The first process that we talk about are the dendrites. These are tree-like extensions at the beginning of the neuron, as you can see, tree-like. And what they do is increase the surface area of the cell body. These tiny protrusions, they receive information from other neurons and transmit the electrical impulse or stimulation to the soma. The dendrite characteristics are they are short and highly branched, and they are receptive or input region and transmit information to the cell body. Next is the axon hillock, a small funnel-shaped structure towards the end of the soma. This plays a very important role because this controls the firing of the neuron. If the strength of the stimulus that is received by the dendrite is strong enough, then only that message will be passed through the axon hillock down the axon. So therefore what happens is, if the stimulus, if the message in here is weak and cannot exceed a certain threshold limit, the axon hillock will not send or pass the signal down the axon. The axon is an elongated fiber that extends from the cell body to the terminal endings and transmits the neural signal. The larger the axon, the faster it transmits the information. Some axons are covered by a fatty substance which is called the myelin. And this myelin will act as an insulator. These myelinated axons transmit information faster than other neurons. We'll talk how and why in a short period of time. Neurons have only one axon. They transmit information, that is, the axon transmits information away from the cell body, always in one direction, from the soma to the axon terminal. They may or may not have a myelin sheath or a covering, and they end in branches that end in knob-like structures in here, and these are called the axon terminals. The axon terminals are the secretory regions of the neuron. These are the ones that will secrete the neurotransmitters that will excite or inhibit the other adjacent neuron. The myelin sheath is a fatty sheet that covers the axon. The sheath is formed in the peripheral nervous system by certain cells called Schwann cells. And this is only true in the peripheral nervous system. The cells wrap themselves around like a toilet paper roll, like this basically, pushing the cytoplasm and nucleus as a bulge external to the sheath. So if this is the axon, they will wrap themselves like this, pushing the nucleus and the membrane to the outside. The portion of the sheath that contains the nucleus right here, as you can see, the nucleus and the cytoplasm right here, will be called the neurilemma. The tight membrane, the inner membrane, so if this is the axon, the membrane out here is rich in a fatty substance called myelin, which is at a higher proportion than other membranes. So therefore what happens is, this inner sheath will be called the myelin sheath. The outer part of the Schwann cell, which has the nucleus and the cytoplasm, will be called neurilemma. The adjacent Schwann cells do not touch each other, but there's a gap in between the cells which leaves a short portion of the axon naked. So as you can see, the axon is covered by this myelin sheath, and as you can see, there's portion of the axon which is naked. These are called the nodes of Ranvier. The sheath is formed by oligodendrocytes in the central nervous system. So once again, this myelin sheath is formed by the Schwann cell in the peripheral nervous system and oligodendrocyte in the central nervous system. No matter whichever cell forms it, the main purpose of the myelin sheath is to increase the speed at which the message or the impulse is transmitted down the axon. Because along the unmyelinated sheath, or the axon that does not have the myelin, what happens is the message moves through continuous wave all along the axon. However, when there is a myelin, now notice the message or the impulse is going to jump from one node of Ranvier to another. So therefore what happens is, this sort of propagation is going to be faster and is called saltatory conduction or saltation. Thus, myelination allows an increase in impulse speed without an increase in axon diameter. Myelin also increases electrical resistance across the membrane. So if there's one axon here and there's another axon here, the message will not be able to pass from one axon to another. So the message will remain within this axon, and it's very highly unlikely that the message will jump through the membrane into another axon, axon 1, axon 2. Thus, myelination helps prevent the electrical current from leaving the axon. The production of myelin occurs in the 14th week in the fetal development. Very little amount of myelin exists in the brain at the time of birth. During infancy, myelination occurs quickly and does not stop until adolescent stage of life. Because of this rapid myelination, it is very, very important that children receive a diet higher in fats than one of an adult. Regions of the brain that contain the dense collection of myelination fibers are called white matter. The gray matter consists of the cell body and the dendrites. At the end of the terminal knob is a gap, and this is called a synapse. So in other words, this, as you can see, the axon terminal that's ending in here, the axon terminal is ending. This is not going to touch the next dendrite. Okay, so here's the dendrite, here's the cell body, and here's the axon. Okay, so this is the axon terminal. The axon terminal is not going to touch the next dendrite of the next neuron. So here's a neuron. It's not going to touch. There's going to be a space. And this space is called the synapse. So how does this neuron 1 communicate with neuron 2? So across this space, there is certain chemicals that are secreted. And these chemicals are called neurotransmitters. So neurotransmitters released from the nerve cells, specifically from the axon terminal right here, carry the signals across the synapse to the next neuron. With this, we come to the end of the histology of the nervous tissue, and I thank you for giving me your attention.